you so much for the kind introduction, and thank you for being here so late. So today I will give you kind of an overview of some of the recently developed methods in our team that are mainly focused on this kind of generative AI that can help us potentially to unlock the complexity of cells. So we are in the midst of genomics revolution that is transforming our knowledge of biology at the cellular level. With advances of single-cell RNA sequencing technologies, we can nowadays measure gene expressions in hundreds of thousands of individual cells. And not only that we can measure transcriptome of a cell, so we can also measure proteins, epigenome, phenome, and with multi-omics technologies, we can even measure multiple of these modalities at the same time in the same cells. And very latest spatial technologies enable us to study cells in their native tissue context and help us to study the fundamental biological processes, such as how tissues develop, from how tissues develop to how cancer cells interact within their microenvironment. At the same time, we are also witnessing an AI revolution. We've seen completely new capabilities of AI system arising with the generative AI paradigm, bringing us to the era of foundation model. So today we think of a foundation model as a model that has learned features that are transferable to a wide range of downstream tasks. For example, most famous examples would be like large language models such as GPT, pre-trained on internet scale data to predict next token in a sentence that can be then applied to a variety of downstream tasks. But how can AI help us to advance our understanding of biology? How can it bring us closer to understanding our cells, how they function, and what goes wrong on a cellular level in disease state? And ideally, our goal is not only use AI to make predictions, but ideally also to help us to discover some new biological phenomena and mechanisms. But compared to the data in natural language processing or computer vision, biological data is inherently heterogeneous. So we collect these data sets from different tissues, different donors, different time points, different species, and also we measure different modalities, for example, multiple aspects of the cells. And the ultimate goal is kind of to develop an integrative, holistic understanding of biology that would capture the complexity of life across different molecular scales. And here, AI methods offer the opportunity or potential to unify these fragmented data sets and extract meaningful patterns that would be generalizable across different biological contexts. And in our prior work, we tackled some of these areas or accesses of complexity. For example, we developed Mars method that was able to transfer annotations across different tissues, Saturn method that was able to transfer annotations across different donors in spatially resolved single-cell data sets. Stellar method, Saturn method that was able to integrate data sets across different species. And also Aging Fly Cell Atlas, where we analyze data sets across different developmental stages. But in this talk today, I would like to start a discussion on how can we gain insights into relationships across different modalities, in particular between the transcriptome of the cells and the spatial organizations of the cells. So for these two modalities, we have two technologies. So on one hand, we have single-cell RNA sequencing technologies that enable us to study cells at unprecedented resolution. But the fundamental kind of problem is that cells are dissociated from the tissue and the spatial context is lost. On the other hand, we have spatial transcriptomics technologies that enable us to study cells in their native tissue environment, but the number of genes that we can measure with spatial transcriptomics technologies is limited. So can we somehow overcome these limitations of these existing technologies with computational approaches in order to get the best of both worlds? So ideally, one would like to have kind of the spatial information about locations of the cells, as well as the full transcriptomic information, in order to obtain a more complete picture of how tissues functions and understand cell-cell interactions. So we started this project with the question, how well can we reconstruct complex tissue structures de novo? So if I start from dissociated single cells, can I be able to predict these kind of complex tissue structures? And we call this process going from dissociated cells to the tissue structures tissue reassembly, because we start from the dissociated single cells, and now we try to organize these dissociated single cells into meaningful tissue structures. But the question is, to what extent is kind of tissue reassembly process possible, given vast cellular complexity, complexity of tissue structures, intricate cell-cell interactions, and also elusive biological mechanisms that may underlie tissue formation? And if it is possible in certain biological systems, how can we do it? How can we learn these underlying rules of complex spatial organization? So good news is that we have lots of existing spatially resolved single-cell data sets. So we started this project with the idea that we would like to leverage these existing spatial transcriptomics data sets in order to learn some useful spatial priors that would help me to learn how cells get mapped to their tissue structures. And in order to do so, what we need to learn is we need to capture both cellular interactions locally as well as globally across the entire slice. So what we need to learn in order to capture these cell-cell interactions is basically how each cell should attend to other cells based on the relevance of other cells to a cell's own locations and gene expressions. And if you think about this, kind of the way we could potentially model this kind of attention is using attention mechanism, which is a kind of key component of today's transformer models. So in particular, we could kind of model these cell-cell interactions in a way that each cell needs to learn how much importance should it give to other cell in its context, and these kind of importance scores would be guided by the learned weights or kind of attention scores. And secondly, we decided to kind of model this tissue reassembly process as a generative process. So in particular, we model it as a conditional point cloud generation using diffusion models. So diffusion models are a class of generative models where the idea is you start kind of with a noise sampled from Gaussian distribution, and then with a number of diffusion steps, the model learns to denoise the data. So in this case, we would start with the noised cell coordinates, and then we would condition these noised cell coordinates on the gene expressions, and then ideally, with the number of diffusion steps, be able to go towards these reconstructed tissue structures. And these two key components, first modeling cell interactions via attention mechanism, and secondly, modeling tissue reassembly as a generative process, bring us to the framework that we call Luna, and this is a work developed by my PhD student Yeast and the master student Shanakia. So in particular, Luna leverages the spatial transcriptomics data sets with ground truth cell locations in order to learn useful priors. So we start with a matrix of cell gene expressions and also ground truth cell coordinates. Then as a diffusion model, Luna consists of two main parts. The first part is a corruption process, where what we are going to do is we are going to sample diffusion time, and we are going to add the noise to the ground truth cell locations. And then in the second step called the denoising process, we are going to learn to recover the original cell coordinates through the denoising process. So basically, we would learn these cell representations using the self-attention mechanism that I introduced earlier to get the cell embeddings. And then from the cell embeddings, we add a fully connected neural network layer to predict the cell coordinates. And now we train the model to learn to denoise the cell coordinates. And in addition, we introduce rotation invariant loss because slices in spatial transcriptomics can undergo arbitrarily rotations and translation of a slice. And using this rotation invariant loss, we don't care about predicting the exact locations, but we care about correctly predicting the relative relationships between the cells. And then ideally during inference, what we would like to have is that we would like to now sample from standard normal distribution, and then with a number of diffusion steps, go towards the reconstruction of tissue architecture. So Luna is linear in time and memory complexity during inference because we use attention with linear complexities. So what this means in practice is that it can infer locations of tens of thousands of cells in just a few minutes on a single GPU. And next, I will show you some results of applying Luna across tasks of different levels of complexity. starting with the first kind of more simpler task where we want to see whether Luna can kind of do a sanity check and evaluate Luna on existing spatial transcriptomics data sets to see whether it can come up with meaningful predictions. So in particular, we tested Luna first on this Murphy mouse brain atlas that has over 4 million cells. We trained the model on 2.8 million cells from data from one mouse. And then we apply Luna to 1.2 million cells from another mouse that the model has not seen. So basically, we take only dissociated single cells from the second mouse. And we see, so here is an example for one slice. Here on the right, I show the ground truth cell locations. These are Luna's predictions, and this is how the fusion process happens in Luna. So in here and here, cells are colored according to the cell classes that are available in this Murphy mouse brain atlas. So we see good performance also across other slices. So this was just an example, but across 11 major brain regions that are identified in this Allen brain atlas, we see that Luna is able to predict locations pretty well. We next looked whether within a certain cell class, Luna is able to actually correctly infer the spatial distribution of gene expression. So here's an example of spatially variable genes for glutamatergic neuron, in particular RXFB1 and RORB, which change their expression across locations. And we can see that even within this kind of certain cell class, the model can recapitulate well how cells change the gene expressions across location. But given the high similarity of the two brains, we next were wondering whether if we exclude a certain cell class during model training, can now model be able to correctly, in another mouse, be able to correctly still predict the locations of this unseen cell class, although it has not seen this exact cell class during the model training. And in particular, to make this task harder, we focus on 34 major cell classes that are identified in this data set, and we exclude one cell class, each of the cell classes. So in particular, we train the model on all cells from one animal, excluding one cell class, one of the 34 cell classes. And then during testing, we use all cells from another animal, including cell classes from an unseen cell class during training. And here is an example for our glutamatergic neuron, where actually Luna correctly positions these unseen glutamatergic neuronal cells with the spatial architecture of the tissue. And if you look at the spatially variable genes, for in particular TSHZ2 and SYT6 gene, which are expressed in different regions of glutamatergic neurons, we can again see that the model has been able to predict. The predictions agree well with the ground truth locations. This is also the case for other unseen cell classes. For example, this is another example for vascular cell class that has different distribution across different slices, and model was able to correctly predict the cells from this cell class, although it has not seen it during model training. So Luna uses a unique method in this idea of learning spatial priors across multiple slices. And existing methods either map dissociated cells to a reference slice, and then they rely on the exact spatial reference. Example of such methods would be a tangram, or they rely on some assumption. So they do kind of this de novo reconstruction, but they rely on some assumption. For example, NovoSpark relies on the assumption that if two cells are in close proximity, they should also have similar gene expression. However, this assumption is typically not satisfied in the complex tissues. So we benchmark Luna using against these existing methods. And here the performance is measured at the Spearman rank correlation coefficient, where we rank cells according for each cell. We produce the ranking based on the similarity based on the distances of the cells and compared to the ground truth locations. And we see that Luna outperforms existing methods by a large margin, and it is the only method capable of kind of correctly inferring these spatial priors. But even if we use the prior knowledge for existing methods about the correct reference mapping, which Luna does not use, even then we see that the model outperforms existing methods. So next, more interestingly, we applied Luna to do the de novo reassembly of the central nervous system single-cell RNA sequencing atlas. So in this case, we don't have the ground truth cell locations. So we still use this kind of model that has been pre-trained on the Murphy mouse brain atlas. But now during the inference, we input single cell RNA sequencing data. And because we don't have the ground truth locations in this case, we integrate the star map atlas with the single cell RNA sequencing atlas, where model has never seen this star map atlas, and we basically use it only for the validation. So these are the estimated now ground truth locations, and these are Luna's predictions that agree well across these 23 different cell classes, and also across, if you look at more finer annotations of 216 cell classes that are available in this data set. And this is again how, so I cannot restart the video for some reason, but this is again how this diffusion process happens in Luna. So if you look at the gene expressions, for example, IQGAP2 gene shows a gradual downregulation across the Emmons hormone region, which is correctly captured here in Luna's predictions. And on the other hand, MIF-TC gene, its expression is minimal within the CA2 region, but it is substantially upregulated in isocortex and olfactory areas, which is captured by the model. And now, because we are now inferring location for single cell RNA sequencing data, we can now actually look for the unseen spatially variable genes that the model has not been trained on, and to kind of identify additional genes that change their expressions with the locations, because now we are inferring actually locations of the single cell RNA sequencing data that has full transcriptomic information. And here is example of applying Luna across different brain regions where we were able to identify these additional spatially variable genes. Luna is also applicable to infer location of spatially unmapped nuclei in the slide text data. So slide text is a technology that enables profiling of single cells and the spatially resolved transcriptome developed by Fei Chen and Evan McCosco. However, the limitation of this technology is that it's very sparse. So basically many nuclei lose the spatial information during the barcoding process. So we asked the question, can we now use the Luna to predict the tissue locations of the nuclei that have been lost during the cell profiling with slide text? We applied across all the data sets available in the slide text paper, but I will show results for the human melanoma data set. So these are spatially mapped nuclei, and these are now nuclei mapped additionally by Luna. So Luna successfully enriched this data set from 4,800 nuclei to the 6,500 spatially mapped nuclei. Now, if we focus on the tumor cells, so in this data set, there are 2 tumor cells that are located in spatially distinct compartments, and they express different genes. In particular, CLU gene is expressed by one group of cells, and this TFP1 gene is expressed by another group of cells. We can see actually that the method correctly positions these cells in these spatially different compartments. And now we used Luna to see if we can now detect additional spatially variable genes for tumor cells. And indeed, we were able to detect additional 400 genes. And if you look now at the pathways that are enriched for these genes, these are indeed pathways associated with tumor cells. For example, the most enriched pathway is MYC targets, which is basically target corresponding to genes that are regulated by MYC oncogene. We can also use Luna to kind of try to simulate what would happen if we exclude certain gene. So in particular, what we do now is like during training, we use all genes, but now during inference, I can kind of simulate the knockout by kind of removing gene one by one and seeing how the predictions of Luna will be affected. Of course, here we cannot say anything about causality, but we can identify those genes that kind of drives Luna's predictions the most. So here we measure basically the performance Luna achieves using all the genes. And now we exclude genes one by one and measure the effect of Luna's predictions, like how much the prediction performance is affected if I now to remove this certain gene. And then we systematically do this across all the genes that are available, and here results across different cell classes we indeed find that those genes that affect the spatial architecture the most are genes that are crucial for cell identity and function. For example, for oligodendrocyte, this is Claudin 11 gene, which is oligodendrocyte-specific protein, and the previous studies have shown that mouse in which this gene has been knocked down show central auditory deficits and neurotransmitter imbalances. And finally, we collaborated with Joelle Lamano's lab at EPFL, where we use Luna now to try to predict the locations of the Parkinson's disease mouse model. So we generated, in Joelle's lab, they generated a Xenium dataset for wild-type mouse on one hand, and then also the Parkinson's disease mouse model was established via unilateral injection of alpha-synuclein preformed fibrils. And then after 3 months, the brain was collected, and we were then interested to see whether Luna would kind of be able to predict the differences between the control and disease sample. So in particular, what we do is we take this model pre-trained on the Murphy's mouse brain atlas, we finely tune it on the Xenium control samples. Model has never seen the Parkinson's disease data, and then we see how well the model can now predict the locations on this data set. So in order to be able to see the differences in the spatial architecture, we take both of these samples and then use Luna to align them to the common coordinate framework using Luna, basically just by feeding both of these samples to Luna. And then we identify cells with locally distinct gene expression profiles between the control cells and a Parkinson's disease mouse model. And then we went to validate these regions using immunostaining against pathological alpha-synuclein in this Parkinson's disease mouse model. And we indeed identify these kind of groups of cells that highly express this protein that is not available in the set of genes that have been profiled, that the model has been able to correctly locate these cells. So while Luna is leveraging this kind of spatial transcriptomics data sets to train the models, next I would like to discuss how can we create these kind of big, multimodal models, even if we only have limited amount of the pair data. And this is the work that has been led by visiting PhD student in my lab, Fabian, and my PhD student, Shuo, and master student, Huyen. So in particular, if you think about existing multimodal foundation models, they require huge amounts of the pair data, like CLIP, or like even bigger models like Gemini or GPT-4o. But they're trained on like tens or even hundreds of millions of the paired samples. But in biomedical applications and biology, we often don't have this kind of millions of pair samples to train these such models. So we asked the question, can we create multimodal models even if we have limited amount of the pair data? So in particular, what ideally we would like to achieve, if you have data sets that you measure in modality 1 and modality 2, it could be, for example, single-cell RNA sequencing, and you're measuring chromatin accessibility, for example, and your goal is to achieve the alignment between these two spaces. Now you can kind of train these models, and ideally you would kind of come to this kind of aligned space. But if you don't have sufficient amounts of the labeled data of paired data sets, what would happen easily is kind of this misalignment. So the question is, how can one guide the model towards a well-aligned solution where basically cells, for example, that are in the same cell state measured across different modalities would align well? So one kind of observation is that we typically have lots of unimodal data sets. So, and then we have also built this kind of pre-trained large unimodal foundation models. And one could kind of think, can we somehow align these pre-trained spaces of these already trained and big unimodal models? So instead of training from scratch multimodal models, can we start with these kind of pre-trained unimodal models and now use maybe more limited amount of the multimodal data to build the alignment between these unimodal models? And there are some existing approaches that do exactly this. So basically, they start with unimodal models that are pre-trained already. So here is just an example of text and visual models. But you can imagine any other different modality. And now what you can do is you can now train this alignment function to go towards the shared space. And this alignment function is typically contrastive learning objective, where you force cells, of the value 4 samples from the same class together, and samples from different classes or dissimilar samples far away from each other. But these existing works still require millions of paired samples. So the question we asked is, can we use less than 1% of the data typically used in the field? Can we use it can we do it much more efficiently in terms of the number of samples that we need to train these multimodal models? So in order to do so, the kind of key idea that we introduce is that we want to be able to preserve the structure in this kind of, from this unimodal space to the multimodal space. So what this means is basically I have already well pre-trained models in the unimodal spaces. So what I can do is I can kind of look at the neighborhood in these unimodal spaces. And now I can kind of impose that I want this neighborhood to be preserved also in the multimodal space. And I can do this for both of the, or multiple of these modalities. So the key idea is that because I don't have sufficient amounts of the labeled data, I want to leverage this kind of well pre-trained unimodal space. And in order to preserve the structure, I can kind of preserve the neighborhood between these after alignment and before the alignment. So we introduced this kind of very simple regularization term that preserves the structure of the pre-trained space in a way that data points that are similar to each other in the pre-trained space should also remain similar in the aligned space. So in particular, how the regularization works, so I start with the pre-trained space and the shared space. I compute the similarity matrices in these spaces. I transform them to similarity distributions. And then what I'm going to do is the minimize Jensen-Shannon divergence to kind of preserve the structure between the aligned and unimodal spaces. Now you can do this, for example, for one hop neighborhood, or kind of you can capture L hops on the similarity graph. And now we add this simple regularization term to any alignment function, your favorite alignment function, which can be, for example, a contrastive learning objective that is used to clip. So we add basically this regularization to both of these modalities. And the nice thing is this can be really easily, it's a very modular framework that can be easily incorporated into existing alignment methods just by adding this regularization. So what we observe now is low data regimes. So now we train the model using much smaller amounts of data than it's typically used in a field. So we train the models using only 80,000 samples, which is less than 1% data used in a field. And we observe that once we add this structure regularization, which is shown here in blue color, comparing to this kind of red color without the regularization, we show that if you do linear alignment on average across different zero-shot classification tasks and retrieval tasks, model achieves 68% improvement using this regularization compared to model without any regularization. And similarly, if you do nonlinear alignment, we achieve 74% improvement across these different tasks. The same improvement exists also across different model combinations. So it doesn't depend on the model you use. So this is an example of using different language models with different sizes. For example, Roberta, different sizes of the llama models. Still adding this regularization helps independent of the unimodal pre-trained space that you start with. And similarly, the results look similar also for different vision foundation models. However, if we look at the absolute performance on some of these data sets, we can notice that despite the advantages of this alignment approach in low data regime, performance is still low on certain data sets. So, but what we show is that if we add only a few in-domain samples, because this is always evaluated on out of domain. So if you just add a few in-domain samples, in this kind of, during the training, this alignment objective, this kind of simple approach can outperform and reduce the gap to large-scale multimodal models that are trained on hundreds of millions of paired samples. So this star here in particular denotes CLIP model that has been trained on huge amounts of the paired data. This regularization also helps if you have less data. So this is an example, scaling down the dataset from 80K to only 1,000 samples for the zero-shot classification, and similarly for the retrieval task. So we still see benefits of adding this regularization, even if you go to the lower, the regimes where we have even a lower amount of the label data. And next, I would like to discuss, can we somehow still like leverage better this like unimodal or even multimodal foundation models to help us actually to tell us what kind of classes exist in the data. And it is a line of research led by my PhD student Artyom and Yulun. Because if you think about this kind of current multimodal foundation models and this kind of typical zero-shot transfer learning paradigm, we still assume that there are kind of categories that are labeled in the data for which we have the labels, which is typically in biomedical applications also often not the case. For example, I may want to use single-cell foundation models pre-trained on the transcriptomics data to discover some rare cell types, and maybe I don't know what exactly categories exist in the data, but I would like to kind of automatically discover the categories that exist in the data from these kind of representation spaces of these big foundation models. So my point here is that this kind of traditional way of thinking about this zero-shot transfer learning is not suitable for kind of discovering these new classes that exist in the data. So the question we asked is, how can we infer these underlying classes in the data without any supervision? So in particular, if I start with these large pre-trained foundation models, and they think about this, very generally, this could be image foundation model pre-trained on real-world images. It could be, single-cell foundation model pre-trained on single-cell RNA sequencing data set. It could be histopathology foundation model. Does not matter. Kind of, it's very general paradigm. So what I would like to do is kind of starting from this kind of big pre-trained foundation model. I would like to use this model to tell me kind of what classes exist in the data without having kind of the labelings of these possible classes. And of course, the trivial solution to the problem that you may say, okay, I can just take these pre-trained representation foundation models and I can run the clustering algorithm on top of these pre-trained representations. But can we do better than that? And we call this paradigm unsupervised transfer because what we would ideally like to do is like transfer in an unsupervised way knowledge from this big pre-trained foundation model. So in order to answer this question, kind of the key insight is that if you think about how we currently often leverage these big foundation models, is that we add a linear head, we just add a linear classifier on top of the pre-trained spaces, and very often like we achieve very high performance. So basically these models generalize really well by just adding a linear classifier. We don't fine-tune anything. So motivated by this observation and the fact that if we measure now the performance generalization error of a linear classifier with agreement of the human labeling. So if I kind of start to kind of randomly permute the labels and now train the linear classifier on top, we actually see that generalization error is lower on tasks that better agree with the ground truth human labeling. And this motivated us, this observation motivated us to develop a turtle framework where the idea is to reformulate unsupervised learning problem as a problem of searching for such labeling so that the linear classifier will generalize well in this space. So in particular, I can start with dataset D. Again, this is like very general. This is just for a simple example, but it could be any kind of dataset. Now I embed this dataset into single or multiple foundation models. This could also be multimodal models or like multiple unimodal models. And now what I can do is I kind of produce the labeling, and now I see how well a linear classifier generalizes in this labeling. And now I basically start optimizing over the labelings to find such labeling so that the linear classifier will generalize well. So basically, I'm searching for a labeling that will have a low generalization error of a linear classifier. So this is a bi-level optimization problem. But searching for a labeling requires solving a discrete optimization problem over all possible labelings. So we resort to continuous optimization via relaxation, where instead of doing discrete search over labeling, I'm going to do continuous search over parameters of task encoder, which is basically a neural network that is producing these labelings. Now, given a labeling that is produced by task encoder, what I'm going to do, I'm going to embed my dataset into multiple foundation models, frozen. I never pre-trained this foundation, never fine-tuned these foundation models. And now I feed the given labeling I split the data into train and test. I feed the given labeling using a linear model, and then I evaluate how generalization error of a linear classifier on the test set. And now my optimization is that I'm searching for such labeling that will minimize generalization error of a linear classifier with a given labeling. So I'm basically optimizing over the parameters of this task encoder. We can also prove that under some assumptions, this procedure will create labelings that maximize the margin of a linear model in the corresponding representation space. So we compared Turtle across a huge number of data sets. So this is a benchmark of 26 data sets from various domains. And we see actually that the method achieves state-of-the-art unsupervised performance. It is available on papers with code. So this is an example from the papers with code on the very frequently used benchmark ImageNet. It's very easy to use the model because you can just start with your favorite foundation models. And the only thing that the model does is basically it trains the linear classifiers on top of these pre-trained spaces. Now we also compare this unsupervised transfer to the standard zero-shot transfer, in particular to the CLIP zero-shot model across 26 data sets that are used in CLIP. So what we observe is, again, CLIP is using this kind of set of categories that exist in the data, while the model does not use any text information. So what we show is that if we run now Turtle in the same representation space as a CLIP, so using only one foundation model, so we achieve same performance. And if we just add another model on top of it, which is very cheap, for example, Dino representation space, so you just add another foundation models, the model actually outperforms CLIP by a very large margin across different model sizes, while being fully unsupervised. So Turtle performance is also correlated to a linear probe. For example, on 5 data sets where linear probe, meaning kind of training a linear classifier, achieves a near perfect accuracy, Turtle closely matches it, which kind of indicates that Turtle could infer optimal classifier without supervision given high quality representation spaces. So if the representation space is good enough to be able to distinguish between the classes, the Turtle could find this optimal classifier. We recently shown that not only that we can do this in an unsupervised manner, this kind of classification tasks, but in collaboration with Amir Zarmir's lab at EPFL, what we showed, we actually proposed a framework called joint inference, where instead of doing independent joint inference, we do the joint inference across to make predictions simultaneously for all inputs in a given task. And now, instead of doing this linear classifier in the inner loop, we would do something like in-context learning in an inner loop. And now we show basically that for example, on reasoning benchmarks, on math reasoning benchmarks, this kind of unsupervised methods can by large margin outperform zero-shot methods, but also can even match the performance of the supervised ICL. Turtle is also applicable to other data modalities. So this is an example of applying Turtle to single cell data, where you start, for example, with single cell GPT or other foundation models. But the assumption in Turtle is that kind of the representation space of this kind of pre-trained model is already like very strong, which we indeed see for some of the datasets of these foundation models, but sometimes this is still not the case. And this kind of opens nicely also my last topic, where I would like to also discuss how do kind of these existing foundation models perform on the perturbation response prediction task. And this is work done by my postdoc Ramon in collaboration with Sarah and Moore's labs. So in particular, we did a thorough benchmarking of perturbation response prediction methods. So we designed a benchmark where we collect approximately 10 data sets across 3 different technologies, including CRISPR-I, CRISPR-A, and CRISPR-Co, and across 5 different cell lines. And we wanted to answer the question, to what extent can we predict the transcriptional outcomes of unseen genetic perturbations? And as metrics, we used standard metrics that have been adopted in the literature, including a Pearson correlation between predicted and the ground truth expression changes of a perturbation with respect to a control cells. So basically, you measure the perturbation effect, you measure the predicted post-perturbation profile, and then you subtract the perturbation, then you subtract the gene expressions of a control cells to see the difference, and then you see how well this matches the ground truth perturbation response. And we designed two simple baselines. So one of the simple baselines is perturbed mean, where the only thing that we do is we just take the basically average of all perturbed cells. So we take the gene expressions of all the perturbed cells as my prediction. And this means that basically for all of my predictions, I will use this kind of same profile for all unseen genetic perturbations. So I will not distinguish between the unseen genetic perturbations. So here I didn't say that we are focusing on the task of predicting unseen genetic perturbations, meaning those perturbations that model has not been seen during the model training. In addition, we designed also another very simple baseline called matching mean for combinatorial perturbations, where we add kind of some priors in the data. For example, if I haven't seen any of these two, like for combinatorial perturbations, we are in particular focusing on, we have a data set where there are two genes that are being perturbed. And if we have not seen any of these genes in my data set, then I will just take the average of all my perturbed cells as my prediction. But if I have seen some of these genes in my trained data set, I can do something a bit smarter, where I can just add the prior that I would compute the centroid of all cells that underwent the same perturbation in the training data and compare, and then take the average to this perturbed mean. While if I have seen both of these perturbations in my trained data, then I would just take the centroid of them as my prediction. So surprisingly, what we find is that for predicting a response to one gene perturbations, this perturbed mean outperforms existing methods or achieves very similar performance. And if you look at the combinatorial perturbations, for example, the most commonly used benchmark dataset is the Norman dataset. We see that this matching mean significantly outperforms as existing methods across all these kind of scenarios of number of seen perturbations. So motivated by this observation, we asked the question why this happens. So why do simple baselines perform so well? So we hypothesized that the reason may be in the presence of systematic variation or kind of systematic differences between the control and perturbed cells. These systematic differences may be, for example, caused by potential selection biases, by the way kind of the set of genes to be perturbed has been selected. Maybe they correspond to functionally similar genes, functionally related genes, or could be also caused by some underlying biological factors. So for example, for replogal dataset, genome-wide replogal dataset, perturbation dataset, this is an example of dataset where there are some underlying biological factors that cause systematic differences. We also analyze different data sets in the paper, but here I'll just show example for the replogal data. So here what you can see is basically that if you look at the distribution of cell cycle between control and perturbed cells, there are significant differences. For example, in the control cell, about 25% of cells are in G1 phase, while in the perturbed cells, about 46% of cells are in the G1 phase. And this can be explained by the fact that p53 positive RPE1 cells with abnormal karyotypes tend to react to chromosomal instabilities that are induced by perturbations through cell cycle arrest. So we next aim to quantify the degree of systematic variation. In particular, what we do is we define average perturbation shift as a vector that points from the centroid of control cells to the centroid of the perturbed cells. And we define perturbation-specific effects as those vectors that point from the centroid of control cells to the centroid of cells that underwent the same type of perturbations. Now what we can do is we can now measure the cosine similarity, and we can do this across all these kind of perturbation-specific effects. And if this kind of cosine similarity is high, this would indicate that this would suggest that all of these vectors are aligned in a similar direction, and also suggesting possibly non-specific shifts in gene expression. So now on the x-axis, we measure the systematic variation across different data sets, across 10 different data sets that we introduced in the benchmark. And we see actually that the amount of systematic variation differs across these different data sets. But for example, Adamson and Norman data set that are frequently used for benchmarking these methods show very high degree of systematic variation. And next, we measure how much this systematic variation is correlated to the Pearson correlation coefficients or performance on perturbation response prediction task. So here, data sets are ordered by amount of systematic variation. And here, we measure the Pearson correlation coefficient. And we can actually see that there is a high correlation between the performance using Pearson correlation coefficient across these different, independent of the method, with the amount of the systematic variation. So what this suggests actually that maybe the commonly used metrics that we use to evaluate this perturbation response prediction task are not the best, and they can be actually influenced by systematic variation and may lead to inflated performance. So instead of, that's why we propose, motivated by these findings, we propose Systema evaluation framework, where we redefine evaluation metrics for perturbation response prediction task. In particular, we suggest that instead of using this kind of control centroid of control cells as a reference, we use the centroid of perturbed cells in the reference in order to emphasize perturbation-specific shifts rather than systematic variation. So basically now we are focusing more on the shifts from perturbed reference, which can kind of capture these perturbation-specific effects. Now, if we reevaluate existing methods using this perturbed centroid as a reference, now what we can observe that this kind of perturbed mean achieves now very low performance, which is what we wanted to achieve. But we also observe that also other methods achieve pretty low performance across different tasks, which kind of indicates that predicting perturbation-specific effects for unseen genetic perturbations is actually a remarkably challenging task. In Systema framework, we also introduce very intuitive metric that we call centroid accuracy, which answers the question, for a given perturbation, is the predicted centroid closer to the actual centroid or closer to the other centroids? So basically, you kind of get your predicted centroid, and then you measure how far this centroid, is this centroid closer to this correct centroid, or it's closer to the other centroids. And the performance centroid accuracy of 1 would indicate that the inferred profiles recover the expected transcriptional effects of perturbations, because the most inferred centroid would be closest to the ground truth centroid. And now we can also use this metric basically to contrast method of your choice with the perturbed mean. And we can define, though, we can kind of group predictions into those that are informative and those that are uninformative. So those, we can divide perturbations in this way. So those that are uninformative are those for which models, like this kind of very simple baseline perturbed mean, also achieves, is kind of better or similar to this, to method of your choice, while those that are informative would be those for which the method is outperforming this perturbed mean. We now use this centroid accuracy first to kind of measure performance across different data sets. We observe that, and here the dashed line shows again this perturbed mean baselines. So we show that we actually observe that on most of the data sets, existing methods cannot outperform this perturbed mean on this centroid accuracy. However, there are some exceptions on Adamson and Norman data set, and in particular, out of all the methods, fine-tuned version of single-cell GPT typically achieves best performance. Now, if we look at what actually predictions, what are those informative predictions, for example, for single-cell GPT model, we see that these kind of perturbations, these are kind of set of perturbations for which this method is better than this perturbed mean. And these are the group of genes that are here are basically mainly ribosomal protein genes that are mainly ribosomal protein coding genes that are highly conserved. So this paper is coming out soon, so it is already accepted, so will be soon published. But the code is already available online, and there we also implement these different evaluation metrics. And finally, I would like to thank my lab members who actually are the ones who did all this work. So this was work done that I presented from mostly almost all my team members. And we also would like to thank our amazing collaborators with whom we had a pleasure to work with. We are also hiring, so reach out to us if you're interested in a postdoctoral position or PhD position. Thank you, and I'm happy to take questions.
Thank you, Maria, for that stimulating keynote. Now we can take questions from the audience. Please move to the microphones. We can also have questions from the virtual platform, so please write if you have any. Maybe we can start with a virtual platform question. I'm going to read it. On predicting cell classes, could Luna predict a space location of cells that have an oncological signal in the RNA-seq, even though the cell class oncogenic cells was not shown in the training?
So we haven't evaluated on cancer data, except this kind of melanoma data set for the slide text. We haven't, I think would be interesting to do the kind of same experiments that we did for Parkinson's disease mouse model, to see basically if we kind of train the Luna on control cells, now fine-tune on the cancer samples, how well or accurate the predictions would be. I assume if it's like highly degraded spatial structure, it would be difficult to expect that model could generalize out of the box if it has never seen anything like that.
Okay, thank you. We can move on with microphone number 1.
Very impressive achievement with Luna model. It's very interesting. You show that Luna outperform all existing models for this kind of work. Did you try Cytoscape that was done by Aaron Newman from Stanford University, by the way, that also doing something similar, and I use it by myself and it's performed very...
So my slides actually are not updated because we added 3 additional baselines in the framework that reviewer asked us to, and we show also that Luna is much better than these methods. So we have additional baselines also incorporated in the framework that it's much more comprehensive benchmarking, but I just didn't update my slide, but we see also still like very high performance benefits using compared to other baselines.
Thanks.
Thank you.
Let's move to microphone number 2. Please go ahead.
Yeah, thank you. Very interesting talk. I have questions about the multimodal like alignment. For example, if you have a modality 1 and modality 2, if your modality 2 is, for example, single cell sequencing, they have very like a sparse latent space. They have like a very clear clustering. But if you like for imaging, like your modality 1, they don't have such like very clear clustering, or like in the latent space, they're like a mass. Then how do you align these 2 modalities actually, like in your common space?
Yeah, so I would say because on both, like for this structure regularization, so you always start with like a pre-trained model, right? So if you are kind of have single cell RNA sequencing data set, for example, you would start with some pre-trained space where assumingly kind of this space is good enough, so your data set kind of cluster decently well, which is the case for single cell data sets, right? And then for the image modality, it's kind of, we do the same thing where you start with this pre-trained space, but even though if your data is not maybe clustering well, if your similarities are preserved, that should be sufficient in a way that if 2 images are kind of close together, and they should be closer than like some other image that is kind of further away. So basically kind of graph or neighborhood is decently well preserved, then it should work. But we never fine-tune the model like these unimodal models. We always kind of, the only thing you train is this kind of alignment function, because in this low data regime, you don't have sufficient amount of the data to do the fine-tuning of your unimodal space. So there is this assumption that kind of your neighborhood similarity in the unimodal space is kind of well preserved.
Thank you.
Thank you.
Okay, we can move to the microphone number 1.
Yeah, I had a quite similar question about structure. I was wondering first, did you use it on biological data? Did you try it on single serial?
No, we are now actually trying, because it was developed with the goal of one other project that we will also soon put online. So it was actually developed with this goal in mind for single cell RNA sequencing data and the images, but we haven't tried it yet for this single cell data, because we submitted this to kind of to NeurIPS, so it's more kind of standard like benchmarking on the machine learning side.
Because the reason why I was wondering is that the method relies on the assumption that you have quite a similar structure across the modalities. But for biological multimodal integration, you would kind of expect that there's something complementary as well across the modalities. Otherwise, you might be less interested in having both modalities if they were perfectly redundant. So I was wondering about your opinion on whether this Platonic representation hypothesis, whether it holds if you were to integrate two purely biological modalities, maybe images in RNA or RNA and chromatin activity.
So, but one also kind of thing maybe to notice here is that we never impose things across modalities. So you basically impose that the neighborhood in the unimodal space should like basically that your neighborhood in the unimodal space is kind of good enough that, for example, for like the single cell RNA sequencing data, your neighborhood is well preserved. But it doesn't have to be the case that kind of in this at least unimodal spaces, this can be like very different, like neighborhoods. But once you start aligning things, then you using this kind of small amounts of the labeled data, then you kind of just impose that in this aligned space, what was similar in one modality should remain similar, and what was similar in another modality should also remain similar.
I agree, but if you preserve the internal structure of each of the two modalities when you project, and you also align the two modalities, if both things are respected, then this means that the two projected modalities have the same structure. Like if you fix their structure and then you align them, then they're overlapping. So I assume you reach a trade-off between the two, but I was wondering.
Yeah, I think this is kind of, I would say, always the case, right, when you do this kind of multimodal alignment, that of course you assume that things can be aligned on top of each other, right, where you may kind of lose some, modality-specific information for sure. But this is kind of the case no matter what kind of alignment function you use, right, because you're always kind of forcing one thing on top of the other to align well in this space, right? But then you may lose indeed some of the kind of modality-specific nuances that you don't capture.
Thank you.
Thank you.
We can take the last question because we are out of time. Please go ahead.
I'm lucky, I guess.
And I'm happy also to stay after the talk if there are additional questions.
Yeah, thank you, Maria, for such a visionary talk, trying to tie together the first two parts. I was wondering if you ever asked the question of whether these rich spatial priors that Luna can learn can be transferred to other spatial modalities that don't reach such high spatial resolution. I'm thinking, for example, of deep visual proteomics, so mass spectrometry-based spatial proteomics.
So we actually tried on the Codex data set, but there we have like only 50 proteins. And it was actually very difficult because the training part is, so what is difficult, because like in spatial transcriptomics data sets, when you have more genes, you can actually measure what I was showing, this kind of how genes, spatially change across locations. But the thing with the Codex, right, or kind of spatial proteomics is that these proteins kind of, there is no distinguishing in model to kind of learn. It would map them to the kind of like very similar location, because the model within a certain cell type, it's kind of difficult to distinguish where these cells should be positioned. Maybe the way would be, I don't know, potentially like to fine tune or something like that on these data sets. We haven't tried, but we tried kind of training from scratch on the Codex, human intestine data, and that was pretty difficult.
Maybe we can talk about it later if you have time.
Thank you.
Maria says she'll be happy to take the rest of the questions afterwards. So let's thank our speaker again for this great talk.